privilege to be able to welcome you all this morning. Thank you for those that are in the room and those that are online. We have an exciting morning planned, and this is the first convening in a while of the Delta Agency Science Work Group. I'm the chair for today's workshop, and I'm just going to go a little bit over the DASW. It is a subcommittee of the Delta Plan Interagency Implementation Committee. Science and policy issues. The DASW gathers science leaders from the DPIIC agencies and other interested parties focused on Delta science issues. And of course, one of them right now is cyanobacterial harmful algal blooms, which is the focus of today's meeting. So I'm going to hand over to Alex Stella the mic so that she can take roll call of agencies and also provide language for public comments.
Perfect. Thank you, Lisamarie. Okay. Members of the public will be provided an opportunity to address the Delta Plan Interagency Implementation Committee, Delta Agency Science Work Group Subcommittee on each agenda item during today's meeting. If any member of the public is interested in providing public comment, please raise your hand during the public comment portion of the agenda item. When called on by the meeting facilitator, you will be asked to provide your comment. If participating via Zoom's online platform, please use the raise hand feature available when you click the reactions button in the Zoom webinar controls bar to indicate you would like to provide comment. When called on by the meeting facilitator, you will be unmuted and able to provide your verbal comment. When done commenting, please click lower hand. If participating by telephone, dial *9 to raise your hand. To lower your hand, dial *9 again. For in-person attendees, please raise your hand during the public comment period of the agenda item you wish to speak on. A microphone will be brought to you to provide comments. There will be an opportunity to provide public comment during each agenda item. The last agenda item is a general public comment period for comments on matters that are not on the agenda and are within the subject matter jurisdiction of the subcommittee. If help is needed to make a public comment, if you encounter any technical issues, or as an alternative way to provide public comment, email engage@deltacouncil.ca.gov or call or text 916-902-6459. The Delta Agency Science Workgroup Subcommittee will be providing subcommittee members, participants, and members of the public the opportunity to participate in this meeting via teleconference pursuant to Government Code Section 11123.5. We will now move on to a subcommittee member agency roll call. We ask that one member from each agency respond to roll call. When your agency's name is called, please state your name, affiliation, and title. Delta Stewardship Council.
Lee, Senior Environmental Scientist, Delta Stewardship Council. Online.
Lisamarie Windham-Myers, Delta Stewardship Council.
California Department of Fish and Wildlife.
I think we can just keep moving through.
Okay. California Department of Food and Agriculture.
Hello, Patricia Bowles, Biodiversity Coordinator with the California Department of Food and Agriculture.
Thank you. California Department of Water Resources.
Ellen Preece, Senior Environmental Scientist, Department of Water Resources.
Thank you. California Environmental Protection Agency. California Natural Resources Agency. Central Valley Flood Protection Board. Delta Protection Commission. NOAA Fisheries West Coast Region. Sacramento San Joaquin Delta Conservancy. State Water Resources Control Board.
Okay. So Meredith Howard, I'm an environmental program manager at the Central Valley Water Board.
Thank you. U.S. Army Corps of Engineers.
Hi, I'm Mick Porter, fishery biologist with the Sacramento District.
Thank you. U.S. Bureau of Reclamation Mid-Pacific Region.
Morning, Todd Plain, Bureau Reclamation Bay Delta Office Project Manager.
Thank you. U.S. Department of the Interior. U.S. Environmental Protection Agency, Pacific Southwest Region. U.S. Fish and Wildlife Service, Pacific Southwest Region. San Francisco Bay Conservation and Development Commission. Perfect. That's the end of roll call. Thank you, everyone.
Great, thank you, Alex. So we'll continue forward. All right, we'll move to the next slide. So the DASW, of course, is the 18 state and federal agencies you just heard listed. They're here in this graphic. And the goal is to engage in education, collaboration, and engagement on topics such as what we're focused on today, harmful algal blooms. The Delta Science Program is a group that many of you have been interacting with, meeting with you, because they've been organizing this meeting. And just to give you a refresher on our mission, it is to provide the best possible unbiased scientific information to inform water and environmental decision-making in the Delta. We have core functions that are particularly highlighted here by this leadership in collaborative science. The other topics that we focus on are priority research, science communication, supporting adaptive management, producing syntheses of current science knowledge, supporting the independent science board, and facilitating peer review. So it's a very broad and busy group focused on improving science actions in the Delta. And I can ask you then why harmful algal blooms, and you might all have different answers to that, but I think it's without question something that we need to focus on. There's concerns that blooms will be increasing in frequency and severity due to ongoing drought and climate change. We've been having HABs in the Delta every year since 2010, and the warming conditions are expected to be part of the recurrence in the future. Right now, there is no HABs monitoring that is consistently conducted or funded. And although there is sampling, which you will see, there is a need for increased coordination amongst the groups that are working on HABs in the Delta. So for the Delta plan, the Delta Stewardship Council does report performance measures, and we have we have one in the Delta plan specifically for HABs, and while we're seeing increases, the performance goal is spatial coverage of freshwater HABs in select water bodies of the Delta to be reduced. And if you go to this website, you can see some of the tracking, the data that's used. This was put together by Scott Navarro, and it shows some of the patterns that have been observed. So it's a place to go. We do report there annually. And additionally, our Delta Science Strategy is a tripartite method to coordinate science in the Delta. Some areas of the Delta Science Strategy do tackle HABs, and they're through they have been through a recent State of the Bay Delta Science installment on HABs along the freshwater to marine gradient in the Bay Delta. And that was published in 2024. I'm sorry, it was published in 2023. And there are 2 priority science actions in the 2022 to 2026 Science Action Agenda. So the first being developing a framework for monitoring, modeling, and information dissemination in support of operational forecasting and near real-time visualization of the extent, toxicity, and health impacts of harmful algal blooms. And the second science action is determining how environmental drivers, such as nutrients, temperature, water residence time, interact to cause HABs in the Delta. Also to identify impacts on human and ecosystem health and well-being and test possible mitigation strategies. This tripartite method focuses on reporting, of course, planning, and focusing on priorities through the Science Action Agenda. So how did we get here today? In 2021, the Central Valley Regional Water Board and the Department of Fish and Wildlife approached the Delta Science Program to facilitate the process of developing a harmful algal blooms monitoring strategy for the Delta. The Delta Science Program hosted a collaborative and interagency developed workshop in November 2022, and a workshop summary is available online with links to recordings of all the talks. There's also a copy in the room. So while developing the monitoring strategy, the authors also worked with tribes and environmental justice experts in fall 2023 and kicked off a community collaboration work group in January 2024. The draft strategy that we are here today to discuss is open for public comment until March 28th. And I'm going to suggest that we're excited to engage with you all about these topics. But from a structural perspective, I want you to understand there'll be, before I introduce the speakers, there will be a, after each speaker's presentation, a 2 to 3 minute informal discussion. And then after all speakers, there will be an official public comment at the end of that agenda item. And so there'll be a 3-minute timer on comments, and we'll just stick with our timeline. But if it goes on, that's okay. But you may be asked to continue offline later. So before we introduce the speakers, let's open up to public comments right now for this agenda item. Anybody in the room have a public comment? Not seeing any hands, I turn it over to Erica.
I see no public comment.
Okay, great. Then let's move forward into our speakers. All right. So this morning we're going to hear from Dr. Berg, Senior Environmental Scientist with Environmental Science Associates. Then we're going to hear from Tricia Lee, Senior Environmental Scientist with the Delta Science Program. And then from Dr. Preece, Senior Environmental Scientist from the Department of Water Resources. These are 3 of the authors of the strategy, and I look forward to hearing their talks. So I'll turn it over now to Mene. Okay, great. Thank you, Tricia.
Thank you. Let's see. Oh, okay. Yeah, this works. Thank you for having me here today. I'll be talking about cyanobacterial harmful algal blooms, or CHABs. CHABs are a growing concern all across the United States. Lately, they've been increasing in frequency, and CHABs are a concern because they produce cyanotoxins. There are a variety of different CHAB species, and they produce different types of cyanotoxins, such as microcystin. This map here shows the risk of occurrence of microcystin toxicity in a number of water bodies across the United States. This map was put together by data from the MERIS satellite combined with field observations of microcystin concentrations. So today I'll be speaking a little bit about drivers of CHABs in general, and also about drivers in the Sacramento-San Joaquin Delta. This figure here is basically a summary of what we know about drivers of CHABs, and they can be divided into two main groups: environmental drivers, which are external to the cell, and then physiological drivers, which are traits of cyanobacteria that enable them to bloom and cause water quality degradation. So in terms of environmental drivers, the main one is drought, which then causes low flow conditions, longer residence times in water, stratification, calm water, warmer water, and also decreased mixing in the water. And especially mixing when combined with cyanobacterial ability for buoyancy regulation, those combine to then pave the way for cyanobacteria to bloom. And I have on this figure indicated with a red box that cyanobacteria, when they bloom, they tend to, or CHAB species, tend to float to the surface. And when they're at the surface and receive continuous irradiance, that's when their growth is the fastest and they can form blooms. In this slide, I am going to go into a little bit more detail about how mixing can promote growth of CHAB species compared to other species of microscopic algae, also called phytoplankton, such as diatoms and green algae. Basically, mixing will increase nutrient concentrations in the water. Mixing can also decrease the light dose or irradiance available to phytoplankton in two ways, actually. One is by mixing the phytoplankton to the bottom where light is lower, and the other is by increasing suspended sediment matter and concentrations in the water column. And so decreased light will decrease growth of cyanobacteria, but decreased light or changing light sometimes increases growth of diatoms. Diatoms are very good at growing under low and changing light conditions. Mixing can also decrease water column stability, which does not affect diatoms or green algae negatively, but will affect growth of cyanobacteria negatively. And mixing can improve conditions for fish and zooplankton, and that can have impacts on the lower trophic levels. So with that primer, I'll turn to talking a little bit about CHABs in the Delta. The Delta is a unique area. It's over 700 miles of sloughs and channels, and it's connected to San Francisco Bay, so it's tidally influenced. The main source of fresh water to the Delta is the Sacramento River, and the Sacramento River has a relatively high sediment load. And when you combine the impacts of semi-diurnal tidal oscillations with riverine flow and wind, you get a relatively well-mixed system with a relatively high sediment load. The dominant CHAB species in the Delta, I'm only mentioning here the 3 dominant ones. There are several others that have been recorded, but the dominant ones are Microcystis, Delicospermum, which used to be called Anabina, and Aphanizomenon. And the top picture in these panels shows you the CHAB species as you would see it under the microscope. And the bottom picture is what it looks like in the water when it blooms. So looking to the left at Microcystis, when it blooms, it looks like somebody chopped up lettuce into very small pieces and poured it into the water. And that kind of morphology is actually unique to the Delta. If you go someplace else and you see a Microcystis bloom, like in Clear Lake, it will look like a continuous surface slick, more like the second panel for Delicospermum. This picture of the Delicospermum bloom was taken in the summer of 2022 near Bethel Island at the edge of Frank's Tract. The right panel shows Aphanizomenon, and an Aphanizomenon bloom looks like grass cuttings poured into the water. And the picture of the bloom that I'm showing here is from Mildred Island. And if you look closely, you'll see some small pieces of lettuce in there. It was actually a combined Aphanizomenon Microcystis bloom that's in that picture. So as I just mentioned, this lettuce morphology of Microcystis is quite unique to the Delta. And here I have 3 different pictures from Delta Microcystis blooms. And they vary from very dense to sparse. So the picture on the left represents a dense Microcystis bloom. This picture is taken from the Stockton waterfront. And the middle is a sparse bloom, and this is from the San Joaquin River. And then on the right, there's an intermediate density bloom, and that picture is from the South Delta. So when Microcystis started blooming in the Delta in a noticeable way in 1999, scientists quickly started ranking the colony densities of the bloom. And a ranking method was developed. It's called the Microcystis Visual Index. It's basically a scale from 1 to 5, where 1 is absent, no Microcystis, and 5 is a high colony density. Basically, the water there will be a surface like continuous density of Microcystis. And so scientists started using this visual ranking early on, as early as 2007 or earlier. And this data set represents the oldest continuous recording of Microcystis colony densities in the Delta. I should mention that it is subject to observer bias because it is a visual ranking. This figure here represents our current understanding of different habitats that promote different densities of microcystis colonies. Basically, scientists have noted that dense colony distributions tend to occur in marinas and dead-end channels where there's restricted flow, calmer and warmer water, and there's a deeper euphotic zone relative to the total depth of the water column. Whereas sparse colony distributions tend to occur in the main channels where you have higher flow. They are well mixed and the euphotic zone is relatively shallow relative to the total depth of the water column. And then intermediate colony densities tend to occur in areas that also have high flow and are well mixed, but they're shallower. So the euphotic zone is relatively deeper compared to the total depth. So as you may have guessed from the previous slides, mixing is an important factor in terms of regulating the microcystis colony density in the delta. This figure here is data that was collected by Preece, who's going to speak next. And it represents chlorophyll concentrations that are due to microcystis bloom. And this shows a vertical slice through the water, going from the San Joaquin River to the Stockton waterfront. So the San Joaquin River is on the left here, and then the Stockton waterfront is on the right where it says McLeod Lake. And so one of the things to note here is that chlorophyll is distributed evenly from the top to the bottom. So this is not like the picture that I showed at the very beginning, talking about the drivers of CHABs, where you saw this surface cyanobacterial bloom. And the reason that the chlorophyll or biomass distribution is so even vertically is due to the mixing. And you also see as you move horizontally away from the epicenter of the bloom, concentrations become, they decrease, they become less and less. And that is because the depth increases. And so the horizontal spread of a bloom is really limited by the depth of the water column and the exposure to light that the cyanobacteria see as they're mixed down. These maps here show stations throughout the delta that various natural resource agencies are monitoring. And on the slide are the microcystis visual index data that's averaged for the last 5 years. So this is basically recent occurrences of microcystis. And the left panel shows the frequency of occurrence of the highest densities of microcystis. And looking at the left panel, you can see that there are actually few spots where you see the very highest densities occur consistently. And they are in the south central and south delta locations, whereas on the right map, you see moderate to low microcystis colony occurrences, and they are throughout the central and south delta. And so part of the impetus for the program that we're talking about today is to understand the drivers that result in high densities and try to mitigate these to such that the delta does not turn into high density blooms all over. And with that, I'm going to conclude. Are there any questions on what I talked about?
2 to 3 minutes of questions in this room.
Keith? Oh, yeah.
This is Keith Gregson, U.S. Geological Survey. Nutrients didn't come up in your talk. Can you speak a little bit about
Yes.
nutrients?
Thank you. That's a good reminder. Nutrients are very important, and nutrient concentrations throughout the delta are relatively high compared to the biomass of phytoplankton, not just CHABs, but any phytoplankton in the delta. And so the delta is a region that we tend to classify as not nutrient limited. And so what that means is that it is a perfect place for CHAB blooms and events to occur. There is, from the nutrient perspective, there is nothing stopping them. Thank you.
Really quickly, Eric, do we have anyone online?
We do. Spencer Fern, you should be able to speak.
Hi, I just had a question about the vertical distribution of microcystis. What was the time scale on that map, unless it's like a total capture of the area?
Yeah, that was basically an instantaneous measurement. And so measurements were made all along that transect, and the measurements were made by sonde. And so the way it works is you would lower the sonde from the surface to the bottom and then move to the next station and do it again. And so probably it's a scale of about 1 to 2 hours to take all the measurements that go into making that vertical slice. So it would be like 2 hours and 1 day. So a snapshot. Does that answer your question?
I guess I mean yes, but was it over? It was just one grab, or?
Yes.
Okay,
Yeah,
then
it was one sampling day. Yeah, so
Gotcha.
it's not integrated over time.
Got it. Okay, thank you.
Any other questions?
Yes, we have one from Zenlin Zhang.
Okay.
You should be able to speak.
Hi, Tricia, I was interested in one of the earlier slides you have about the mixing, the impact of mixing, like how it just decreases HABs but increases other type of diatom. I'm just wondering what is the different characteristic of HABs that result in this like different response to mixing?
Yeah, there are basically 2 main characteristics. In order to be able to photosynthesize at very high irradiances, you need to have photoprotective mechanisms, and cyanobacteria have a variety of different photoprotective mechanisms, including different accessory pigments. There are various theories as to what role the cyanotoxins play in the ecology of cyanobacteria. And one hypothesis is that the cyanotoxins provide photoprotection to the cyanobacteria. And so as cyanobacteria become light stressed, they will produce more cyanotoxins. That is one of several theories. And then also cyanobacteria, some of the species that form the CHABs have pretty low photosynthetic efficiencies. So they actually need to be exposed to continuous highlight. And that is a major difference relative to other species like diatoms that do better in changing light and low light.
Thank you. That makes a lot of sense.
There's no
Any other
further
questions?
comment. There's no further comment.
Oh, okay. All right. Tricia.
Okay, thank you, Monet. So I'm Tricia Lee. I am going to be bridging between this introduction to HABs and where we went with the strategy, the monitoring strategy for cyanobacterial harmful algal blooms by talking about knowledge gaps and collaboration opportunities. So Monet just had this slide up earlier. We have this frequency of microcystis that's commonly seen using the microcystis visual index, which she described. And although microcystis, as she also described, is just one of many potential harmful cyanobacteria that we see in the Delta, it's a useful example for how we can strategize data collection to better inform cyanobacterial harmful algal blooms. So on the right side of this slide, we see that there's these three major water quality sampling efforts that go out, and they go out on different sampling frequencies, and they collect discrete water quality samples. In this case, we're showing where they collect discrete samples for phytoplankton ID. And we can see in this microcystis visual index figure on the left, there's this high frequency area for microcystis visual index prevalence. And there are some sample locations, discrete sample locations that overlap with that, but there could be a lot more directed data collection to target this high prevalence area for cyanobacterial harmful algal blooms. So this is an example of a need for potentially an opportunity for us to improve coordination and collaboration to improve water quality data collection. And we hope to do this through implementation of the cyanobacterial harmful algal bloom monitoring strategy. So harmful algal bloom monitoring is not a core function of any long-term water quality monitoring program. And as Monet laid out, we do understand the major drivers for bloom formation in the Delta, but we don't have detailed information about status and trends for cyanobacterial harmful algal blooms and drivers on a site-specific basis. And this is the type of information that we would need to have to be able to adequately implement mitigation in different sites or forecast cyanobacterial harmful algal blooms. So through the workshop that Dr. Windham-Myers described in her first talk, and we defined some data and collaboration gaps with the community, and then the authorship team has been working to expand on those data and collaboration gaps, which all of these can be found in the monitoring strategy, the draft monitoring strategy. And some of these data and collaboration gaps really build off of each other, and we feel that through addressing them through the implementation of the strategy, we could help to close some of these gaps. So, for example, better coordinated agency data collection for harmful algal bloom, for water quality data that would inform harmful algal blooms, data collection. This would help to address the data gaps to inform dynamics and mitigation strategies. And additionally, if we were to standardize methods and analyses for cyanobacterial harmful algal bloom data collection, we could also improve data comparability across different sampling efforts. As mentioned, we do know that there are what the general drivers are for harmful algal blooms, but less understanding of specific changes in the environment that could impact the development of cyanobacterial harmful algal blooms, such as site-specific processes and climate change, and also the specific interaction of drivers to cause cyanobacterial blooms, which Dr. Berg also laid out. And this also ties in this sharing of data. If we could also improve sharing of data, would help close some of these collaboration gaps on making sure that data is available publicly and improve on this collaboration gap to expand really collaboration between interested parties and communities who may be interested in the data that is produced. And an overarching major data gap gap in this effort is that we do recognize that there's not enough information or funding to realistically implement these any mitigation measures to address harmful algal blooms or to forecast cyanobacterial harmful algal blooms in the Delta. So this is the information that really laid the basis for developing the goals and objectives and recommendations for the draft strategy, which Dr. Ellen Preece will go through in the next talk. I will just open it up for any discussion on this topic. If there are questions or comments from the in-person audience, we can take those first. Eric, do we have anyone online?
There's none online.
Okay, I'll pass it to Ellen Preece.
Hi, everyone. So we heard about an overview of cyanobacteria blooms in the Delta, some data gaps that we have identified, and now I'm going to go through the goals, objectives, and recommendations at a high level of what's in the strategy. So we developed this schematic to very high level. This is like a snapshot of what is in the strategy. So we defined the problem that we need this collaborative and cohesive Delta CHAB monitoring strategy based on the available scientific data and the known collaboration that's already occurring. We identified data and collaboration gaps that Tricia just described. And then we defined the goals and objectives, and that's what I'm going to spend my talk discussing. And at the end, I'm going to suggest a few ideas for implementing the multiple recommendations that we have in the strategy. But we also wanted to recognize that this strategy is written in a specific time with the knowledge that we have now. We're going to learn more, and the science is going to need to adapt. And as that's going to lead to the need to come up with new recommendations in the future, and perhaps to shift the goals and objectives that we present in the report as we learn more. So we defined 5 goals for the monitoring strategy. The first was to enhance Delta CHAB collaboration. The second was to identify monitoring questions, goals, and objectives. Goal 3 was to develop a Delta CHAB monitoring program. Goal 4 is to develop collaborative reporting protocols. And goal 5 is to utilize a data sharing platform. Now, the next few slides, I'm going to dive deeper into each of these goals and the objectives that are associated with them. So goal 1 is to enhance Delta CHAB collaboration. The first objective we have here is to organize a collaborative approach to implement the CHAB strategy. We recognize that no one party or entity is going to be able to lead this effort, so we need to have a collaborative effort in order to implement it. Objective 1.2 is to promote coordination, collaboration, and communication among agency and community partners. There are a lot of people that have an interest in CHABs, and we want to include everyone in this strategy. Objective 1.3 is to identify mechanisms to ensure the sustainability of long-term CHAB monitoring and collaboration. So, we recognize that this is going to take a lot of effort, but we're hoping that this is the start of this long-term program. Now, there are many recommendations that fall under each goal, and I only have a short period of time to speak, so I'm going to give you one example recommendation. So the recommendation example for goal 1 is to hold an annual meeting focused specifically on Delta CHABs. And this is to capture the different needs of various agencies, dischargers, tribes, partner organizations, and other partners. The monitoring strategy will need to be implemented in a collaborative and coordinated manner. We think hosting an annual meeting for Delta CHABs will allow this ongoing coordination. So some of the things that we mentioned in the strategy is identifying someone that will need to chair or co-chair this meeting, perhaps on a rotating basis, because it will take time and effort to organize, and it'll also require a little bit of funding to reserve the room and such. Ideas that we have for the annual meetings is to share data and information that's been collected over the past year. We think it's a good opportunity to have a training session. And so that can be so that we can have coordinated monitoring efforts amongst all of these different people. So for example, you heard about the Microcystis Visual Index and the subjective nature of that data collection. Well, at these meetings, we can have a training and try to be more coordinated in how we collect the data so that it'd be more comparable across people that collect that data. Goal 2 is to identify the monitoring questions, goals, and objectives. So things to consider for this is how will monitoring results be used by decision makers? Objective 2 is to consider the data and monitoring gaps needed to answer management priorities. We could all come up with a number of different questions that we want to ask, and different agencies or groups may have different priorities. So working together to kind of organize those priorities and determine which ones we can answer in a more short-term area will be useful. So this is the example recommendation for goal 2. There are a list of about 6 management questions in the strategy. These are just draft management questions, and they were developed by the State Water Board, the Central Valley Water Board, CDFW, and DWR. This is not meant to be an exclusive list. This is just a start, and so we anticipate that this will be built upon through the strategy efforts. The example category we have here is status. Some other categories that we identified were trends, mitigation, predictive models, or predicting when blooms would occur. Then we have a column for the management question. The management question here is, what is the overall magnitude and spatial extent of CHABs within the Delta region? So filling in those gaps of, we showed you there's a lot of areas that are not currently monitored. We need to have a better spatial coverage so that we can answer that question. Examples of decisions that are supported by the monitoring. We want to prioritize water bodies or hydrologically distinct areas, and then inform status and trends reports to the public. So I first read a large regional spatial scale management question, but you can get even further into the details. So a small localized monitoring scale question is, are CHABs degrading water quality in a specific area of the Delta? When is that degradation occurring? Can you narrow it down to a couple months out of the year or not? And then decisions that could be supported by the monitoring, maybe public health advisory posting, letting the public know that this area of the water should be avoided for some period of time. Another idea is that it can form changes to compliance monitoring. Goal 3 is to develop the Delta CHAB monitoring program. So objective 3.1 is to identify specific monitoring programs needed to achieve the management question and goals. I want to point out here that we don't think that a single monitoring program is going to answer all of the goals and questions. So we're probably going to need multiple monitoring programs. The next objective is to identify priority monitoring parameters, locations, sampling period, how frequently you're going out to collect the samples, and then what methods you want to use to collect your data. The next objective is to create implementation guidance for Delta CHAB monitoring. So once again, that's making sure we have this cohesive effort and that data can be comparable. And then objective 3.4 is recognizing that there are a lot of efforts that are in place right now. There's a lot of data being collected. So how can we synergize with that data to leverage ongoing efforts? So the example recommendation for this goal is recommendation 3.1, based on the goals and objectives developed in goal 2, identify monitoring programs and special studies needed to achieve outcomes. I'll just point out there are 19 special studies. I'm not going to get into them today, but they are listed in the strategy. But what we wanted to do is give an example of how to design the program. And this is very high level. This will require a lot more detail than the couple of bullet points. But the management question we chose for this is, which hydrologically distinct areas of the delta are at risk of experiencing CHABs? So you need to think about what kind of geographic sampling you need to answer that question. So possibly to identify the locations known for having the CHABs or the most severe CHABs. So that could be like the marinas and the dead-end sloughs that have those dense harmful algal blooms. Identify locations that may be prone to CHABs based on hydrologic characteristics. So maybe you're picking out shallow areas or peripheral areas of the delta that are not as deep and don't have as much mixing. You next need to consider the spatial, temporal, and how frequently you're out there sampling. How often do you need to be out there to make sure that you're capturing bloom development? A lot of sampling is discrete samples, and blooms are transient and episodic, and so you may miss a bloom depending on how you space your frequency of sampling. And then you need to consider how many samples do you need to represent a specific area. Metrics, select relevant driver data for the chosen site. So you may not be able to collect everything everywhere. What's the most important data to collect to answer this specific question? For data, do methods and SOPs meet data management and quality control that can then be used for decision making? So you want to make sure that the data that you're collecting can ultimately be used by decision makers. They have enough confidence in the way the data is collected. And then additional considerations, how much data is needed to inform that management decision, and how can we leverage this with other ongoing work? Goal 4, develop collaborative reporting protocols. Want to validate and standardize current methods used for monitoring CHABs. This goes beyond the visual index data. Also want to make sure, for example, phytoplankton ID, are the results comparable between laboratories? Are different laboratories identifying different species or different percentages of different species occurring? Because if they're not comparable, it's really hard to compare samples taken from different areas of the delta. And then develop protocols for accurate and timely reporting. Recommendation example here is to compare, review, and standardize sampling and laboratory methods to ensure that we're having this consistent and comparable data. And this is tricky, I think, just because different agencies have different rules in place on how they want to collect data, how they want to share data. So how can we work together to try to make data more available to everyone? And then the last goal is to utilize a data sharing platform. Our objectives for this goal are to identify existing CHAB and HAB data repository platforms. There are a number that are already out there. Explore how to integrate Delta CHAB monitoring data with these existing data repositories, and then develop protocols to make the data accessible and available to all people so that it's not hidden in some strange place on the internet that no one can find. And the example that we want to provide here is coordinate with the NOAA MIRHAB data dashboard. And just quick background, NOAA funded a monitoring and event response grant to the San Francisco Estuary Institute, the USGS, DWR, and numerous other partners. And that started in October of 2023. And the goal of that 5-year project is to increase HAB monitoring across the entire San Francisco estuary, so not just the Delta, But one of the main goals of that project is to develop a data dashboard, and the San Francisco Estuary Institute is leading that data dashboard. And the idea is that they're going to take a lot of the data that's generated and put it into a single data sharing platform. And this table is from the grant proposal and just gives an overview of the types of data that we are going to include in the data dashboards, where this data is collected, and then the type of data. We've coordinated with the San Francisco Estuary Institute to talk about how we can include data that's generated from the Delta HAB strategy into this data dashboard. They're very excited about being able to include all of that. So, you know, it may just be, part of it may be a page that links all of the existing web pages that address HABs in the Delta into a single location. So you're not scouring for different places in the web trying to find your answer. This will consolidate it into a single location. Hopefully to have some visualizations and other useful information on the page so that people can better understand what the data means. This project is only funded for 5 years, but that gives us really good opportunity to at least get this set up. And the goal of the NOAA MURHAB project is to find long-term funding to sustain this. So hopefully, you know, we'll have the product in hand over the next 5 years, and then we will continue to be seeking additional funding to keep that going. And so we can all utilize this single location. And my last slide is, how are we going to implement this Delta CHAB strategy? I think that's going to be an ongoing effort. We don't have the complete answer to that question today, but some ideas are leveraging the MURHAB project that I just described. The State Water Board is funding the USGS to develop at least a monitoring design for some CHAB monitoring to answer some of their questions. The Delta Stewardship Council will continue hosting collaborative work groups so that we can keep implementing discrete pieces of the strategy. And then we identified 19 special studies that various groups could choose to lead as they find funding to implement those studies. And that is all that I have today.
Questions from the room or suggestions or ideas for Dr. Preece? Good question.
Yes.
Thank you for that presentation. Can you give a couple of examples of what the 19 special studies are? Maybe one or two that rise to the top of your brain right now?
Yeah, I think we talked about some lab-based studies to compare like different detection of toxins. There's just different analysis methods out there that can detect toxins, and it's unclear if they're completely comparable. And so that was some ideas. We have the State Water Board, when they wrote the FHAB strategy, had a number of recommendations of how to utilize remote sensing in the Delta. And so, you know, we build off of those suggestions so that we try to implement more remote sensing for early warning. We said a special study could be developing a predictive model for a specific area of the Delta, possibly using machine learning or AI as an idea for that. And so it's rising to the top of my head right now. Yeah. Mitigation. Yeah. Oh, is that funded for sure? Okay. Do you want to mention it?
So one of the other studies, I think that's in there, right, Ellen?
Yes, that is a special study.
So we're going to fund a study through SWIRP to basically convene a technical advisory committee to basically, it's kind of a paper exercise, but kind of make some recommendations as to what mitigation methods or approaches would be most appropriate for the Delta. And that is, we're starting to work on that contract now, so a contract wouldn't be executed before like probably mid-year of 2025. But I think that was one of the things is like, you know, give us a little bit of a roadmap, because there's a lot of different types of mitigation options out there, and there's a lot of constraints in the Delta when it comes to permitting. But it's also just like, well, what's realistically appropriate that might work? And then we could maybe fund some pilot studies down the road. So that's maybe one that we're going to fund. well, it's not maybe, we are funding that. But I think that's a good example of where if we lay out all of these ideas in a document, then you have different agencies that can kind of come in and pluck one of those off as a piece.
Thank you. Yeah, that's a perfect example. I believe that's one of the last special studies. So we grouped the special studies into different categories, like lab-based, there's some field-based ones, there's the mitigation-based ones. And what Meredith just described is exactly what we were hoping for. And of course, it was in parallel. It's not, she was already working on it prior to us, handing the strategy. But the idea that people can choose one of the special studies or part of a special study and advance it on their own as funding becomes available. And the mitigation one is a great option because everyone, that's why we're here, is to solve the harmful algal bloom issue. And a lot of the mitigation strategies out there are for lakes, and those are not applicable to the Delta. And so what can we realistically use in the Delta?
Thanks. My name is Henry DeBay. I'm the Deputy Executive Officer for Science at the Council. I just wanted to take this opportunity to highlight that the Council is planning to release a notice or a call for research in the coming weeks. I know we've been saying that for a while now, but it's actually going to happen. So, if there's anybody in the room or listening who has interest in, taking some of these special studies and putting them forward as applications to the research call, there's an upcoming opportunity. So more to come. If you're on our listserv, you'll get a notification for when the call is available and what it needs to respond to. But in short, it needs to help implement the Science Action Agenda, the 2022 to 2026 Science Action Agenda, and CHABs is a focus in there. So I think there's definitely some connections to be made. So yeah, stay tuned for that call.
And I know we've used these documents, like the FHAB strategy personally has helped us get the NOAA funding. So being able to pull from these established documents is useful for that type of funding or federal funding to fund these different projects. So we hope people will be able to use the strategy for that purpose.
We have two. First one will be Spencer Fern. You are allowed to talk.
Okay. Hey, Ellen. I had a question about maybe timeframes, and I know there's no time frame currently set for this strategy, but say we have a monitoring strategy in place and then something changes. I know you alluded to being able to adapt to science and then being able to make changes as necessary. If there was a need to make a change to a strategy plan or make a alteration due to maybe something from community involvement or the tribal nations, whatever happens to be a recommendation, is there a turnaround time where it would be adapted to a statewide monitoring agency, or what would be the steps to making the changes? And I know that's a hard question, but I'm just curious.
I'm gonna have Tricia answer that question.
Thanks, Spencer. So I actually think that the way the strategy is written, it can be used as a living document of type. So a lot of those recommendations, when we learn, we wrote a little adaptive management section at the end of the goals and objectives. And as we learn more, I do think that we can take that into consideration and use it to inform an additional round or an additional phase of kind of conducting the same steps. So that might not be applicable for all of the goals, but definitely goal 3 that talks about developing a monitoring program. I think that definitely could be done in a stepwise or phased implementation as we learn.
Okay, thank you.
And our next speaker is Zenlin Zhang. You're free to speak.
Hi, Alan. I'm really interested in your proposed plan to coordinate effort between different monitoring programs. My question is, as I'm aware that there are a lot of environmental programs that's already monitoring environmental data in the Delta, like EMP or IEP, and also I think CDM data as well. So I'm just wondering, how coordinated this existing effort with the HABs monitoring? Do they tend to measure at the same location and time, or they're like completely independent program?
I think it probably depends on the parameter. Oh, I'm echoing. But for example, I'm thinking of the figure that Tricia showed, which was the phytoplankton ID. And as an example, DWR, the dots on that map, they're out there monthly collecting those samples at some of those locations. I know that CDFW was also on that map, and they're collecting, I don't know the exact times that they're collecting the samples, but they're doing it in response to their fish. And so they're basing sampling around fish. And so it may only be a few times during a certain period of the year. And I showed someone that the map, I don't recall who it was, and they said, oh, I didn't even know CDFW was collecting those phytoplankton samples. And so I think that to me really highlighted how some people know that this is happening, but a lot of people don't. And so how can we better be aware of what everyone is doing, and hopefully that'll be one of the outcomes of this strategy.
Thank you.
The next person is Christine Job. You are free to speak.
Yeah, hi, Ellen, I hope you can hear me. Okay, great. I have a multi-part question. This relates to one of the slides that you had shown. You were talking about decision makers, and I wonder if you could just, I'll read my questions I'll go through my questions first, and then hopefully you can provide some insight. So I was wondering if you could kind of elaborate a little bit more about, like, who are these decision makers that you kind of referred to in the slide? Have they also been brought into the conversation that you've been having with other individuals about, collaborating with all of the other groups? And if so, what kind of information have they sort of been providing you in regards to what kind of information they would be needing to, make some sort of decisions? And in regards to the decisions, what kind of decisions might those be?
Okay, well, the easy answer for me is the state water board. And okay. So they are regulators. And so I'm thinking as this example of regulatory decisions that could be made. And there's a variety of different things they could do, which, for example, requiring certain water quality objectives in, say, some locations of the Delta, or using flow or other parameters to manage water quality. As an example, the State Water Board has been very engaged in this. We have opened it up to all agencies to be engaged, and there is a smaller working group that has been involved in the planning of this, and that we've had, multiple discussions with about how they could use this data. And so I think, like, the State Water Board, or some divisions of the State Water Board, are very interested in, say, like a predictive model. And so that would be of high interest to them. I think other agencies and decision makers, that's lower interest, but I think they would like to use a predictive model to be able to determine where and when HABs are going to form, and then make their regulatory decisions based on that. Well, I'm not speaking too much for them, but I think that's kind of a high level overview of, like, one group that would be a decision maker. I think CDFW could be a decision maker for fish, depending on what their specific or priority questions may be. And they have also been engaged. Karen Odkins is a co-author of the strategy.
Oh, can you still hear me?
Yep.
Oh, okay, great. I was wondering about decision makers in the sense from, like, maybe the local communities that are impacted from a lot of the CHABs. You mentioned that some of the densest blooms are seen in areas by marinas. Those are areas where there's, a commercial sector usually involved. So I'm just wondering if, the local community supervisors or developers, or, any of decision makers associated from non-governmental agencies are maybe being brought into the conversation in regards to CHABs?
I think to an extent, for example, we met with the Delta Stewardship Environmental Justice Board. I think it was in November, and talked about the strategy and get their input into this. And I don't know, maybe Tricia, you have some, also talk about who else you've reached out to.
Yeah, we definitely are interested in collaborating with local communities and working with those individuals. We haven't had a lot of interaction with those individuals, like with those local agencies yet, but we are definitely open to it. Yeah, if there are connections that folks have that they could share, that would be excellent.
Okay, thank you.
If we have, you know, can start having this annual meeting and getting the word out and getting those people engaged through that platform at a minimum, then they can be more involved through the process as it keeps evolving.
Great. Yeah, I think it's important to make sure that that portion of the community, one, you know, is made aware of the efforts that are undergoing right now with the strategy that you're trying to develop, so you can bring them sort of early into the process, even if they're not ready necessarily to fully engage at this point, but just making them aware that something is kind of underway so they can maybe plan for their engagement more intensely at a further time period. But I'm just thinking, because of the sense that there's always this idea that the government needs to solve everything, and we're dealing with biological species here where we have a lot of not necessarily control over some of the drivers that you mentioned in your presentation. So some of the ways that people may have to consider mitigating might be based more on how things are developed or where they're developed, and having that understanding, right, of if you're developing in a certain area, that there's a higher risk possibly for having these kind of blooms occurring in those waters. So again, it's just more awareness and getting involved and seeing where they can connect where possible. Thanks very much for the information you shared. I thought it was great.
And just a couple add-ons. There have been some NGOs that have participated in this effort as well, and have been at the initial meeting and then at these smaller group meetings. But if you have like a list of people that you think may not know about this, but should, if you send it to Tricia, she would be happy to distribute the information and add them to a listserv.
Great, thanks, I will, thanks.
There's no further public comment.
Thank you.
So at this time, we're gonna go to a break. I think we will shorten it a little bit to a 11-minute break because we are a little bit behind schedule. But so we will okay, go ahead. So let's go ahead and take this one last informal comment or question.
Barbara, go ahead and talk.
Okay, the unmute worked. Can you hear me okay now?
Yes.
Great. Hey, good morning. I kind of wanted to talk about the last comment and talk about the whole issue on kind of outreach and standard setting. One of the reasons you have difficulties with local engagement is because public health departments claim that they are under no obligation to report this information out. And if it's not kicked up by a public health department, the counties will not step in. They will say it is a state issue. So we're stuck in a circle, and we're stuck in a circle that needs to be broken. And we need a numeric CHABs standard that could then be translatable to public health departments so that they do their thing on notification, and then would kick in community engagement. The idea about blaming development of anything in the Delta, considering there really isn't any interior development going on in the Delta, to me is a bit of a red herring. And the reason why is that before 2010, I mean, these places were there. They were there historically, and the marinas were there historically. So we really have to get a standard set in place that kicks local government into doing things to notify people, to protect public health, and to participate in mitigation, because this is such a multifaceted issue. It's local cleanup in some cases, it's what water is coming through, it's climate change, it's all of the above. So I think that's really important to keep in mind. There's a long history in the Delta of kind of blaming the community for things that it really has no control over. And so again, we have to have that standard set, I think, to be able to make that change for the kind of notifications and broad participation. Otherwise, you're not going to get it out of local government.
Yeah, thank you, Barbara, for that comment. I also want to remind everyone that this earlier portion, and that was a very astute observation, and I think I want to remind folks that the official public comment period will be after the 15-minute break. So if you want to reiterate your comments as a public comment, please do so at that point as well. But thank you, Barbara. I think I'm going to send us to a break now. Let's break until 10:35. Started with the rest of our morning. So I have just one more section before we start the public comment on this agenda item. So refresh everyone, bring everyone back from break. The timeline on this, we had hosted a harmful algal blooms monitoring workshop in November 2022, which many folks in this room and people online also helped out with. Over 2023, the authorship team has drafted the strategy that is out for public comment now. And we also did start this work group kickoff in January 2024, in which we assembled technical folks and community members and interested parties to hear from them on the goals and objectives. We plan to keep those work groups going in the future. At this time, we have the draft strategy released, and the public comment period is open until March 28th at 5 p.m. And our next step from here is to present this to the Delta Plan Interagency Implementation Committee, of which this Delta Agency Science Work Group is a subcommittee. So the Delta Plan Interagency Implementation Committee is on April 15th. We will be presenting on the elements of the strategy, similar to what we did today. We'll also share any major feedback and edits that we received from the public comment period and also from the Delta Agency Science Work Group. We will also be asking the DPIIC for their endorsement of the strategy. And beyond that April 15th ask for endorsement, the additional next steps that we would like to go into is to start to implement some of the recommendations from the strategy. So when the strategy is completed and any edits are incorporated, we really would like to start working towards implementation. And some of the first steps up would really be an opportunity for members of the public, members of the community, different state agencies and federal agencies to work together with us to start to really to have those work groups to start to implement pieces of it. And so some of the first things that we would implement are to identify co-chairs or a mechanism for someone to lead the coordination and implementation of the CHAB strategy, and also to form this advisory committee to develop the final goals, questions, and monitoring strategy. We do plan to work closely, as Ellen had mentioned, with that MRHAB. They have already assembled a management and technical advisory group that we think this advisory committee could be very closely related to, so that we are building off of work that's already being done and not recreating work. So we will be starting to, after this, if the strategy is endorsed by DPIIC, working group will be convened by the science program shortly after to implement some of these first recommendations. And we do hope that people reach out and are able to join these working groups in order to help share the responsibility of addressing harmful algal blooms. So this is not yet the public comment period. With that, we can take any discussion on this item. I also want to remind everyone that the draft is currently posted and we are receiving comments until March 28th at 5 p.m. And we can take any in-room questions on next steps.
My name is Ivan from Restore the Delta. I'm the Deputy Director, recently started. So in the next steps, I very much like that this is moving quickly through April to DPIIC to get their vote and their discussion on it. Just kind of looking out a little bit further, what does implementation as well as kind of your idea of community outreach look like, Tricia?
Thank you for that question. So we have some other elements that are kind of underway already. So Ellen mentioned a little bit before that State Water Board has already funded a project with USGS to develop a monitoring design for harmful algal blooms. And we'd like to use these work groups. I'll just toggle backwards, even though there's a ton of text on this slide. We'd like to use these work groups as an opportunity to receive community feedback on all of the elements of implementation. So these will be open to, they won't be public meetings like this is a public meeting, but they will be open to interested parties to attend and be able to provide any input on how the implementation of the strategy is done. Any other in-room questions, comments? Eric, do we have anyone online?
No one online.
Thank you. Okay. So before we go to public comment, not just on this next steps, but on anything that we've presented, CHABs monitoring strategy, are there any other questions or discussion that people would like to get into at this point? Eric, anything online?
Nothing online.
Sorry, our mics might be too close together. Okay, so at this time, we would like to move to the public comment item. This is the official public comment item. It's being recorded, so we will take them into account as we move into finalizing the strategy. For those of you just joining us, we will provide a 3-minute timer for each contributor. If you exceed the timer, you may be asked to continue the conversation offline. Reminder that written comments are being accepted. And for any public comments on this item, I'd like to now turn to our in-person audience to see if there are any in-person comments.
This is Ivan Sienok, Deputy Director at Restore the Delta. I have several comments as well as kind of discussion points to point to agencies in the room. First of all, Restore the Delta thanks everybody for the hard work in bringing this together. Harmful algal blooms is important for the community we serve and that we try and outreach and advocate for. So first of all, outreach is critical to communities. This needs to start quickly. Restore the Delta is here in the room. Restore the Delta is on these committees and engaging with every agency in the room to really ensure that advocacy reaches the communities. But we can't be the only ones. We work with other nonprofits. We work with local community agencies, as well as local stakeholders. Those people also need to be brought into the room because if they aren't signposted, if they aren't notified that a numeric standard is coming, public health sectors won't be prepared, local agencies won't be prepared, local governments won't be prepared, and they need to be brought up to speed. Because this is a community effort, thanks to the agencies doing larger work, but this outreach is critical. Secondly, the records and the distributions of these meetings and informations should also be made available for an issue that Restore the Delta has come to grapple with for the advocacy that we do, is that records need to be posted because most of the community members are working 9 to 5 or 7 to 6 or during hours where they cannot attend public meetings and they can't sit here. So for advocacy and outreach, it is critical to record these and to send these out and make this publicly available so that if they do seek to provide public comments or they can't get a hold of any of the advocacy groups like Restore the Delta, that they can learn and engage with this process as well. Because these are the communities that live next to McLeod Lake. My office is 2 streets down from McLeod Lake, so I know when it gets really green and slimy. But these are also communities that farm, fish, and live next to these HAB outbreaks, and that is critical.
Thank you for your comments. Eric, do we have any comments online?
No comments on it.
Okay. Final reminder, we're still accepting written comments as well until next Thursday. So please do submit those. You can find that information on the Delta Council webpage for public comments that are items that are currently posted for public comment. And I am going to actually go ahead and pass it over to Lisamarie for some other announcements.
Well, thank you all for participating in that discussion, and we look forward to your comments submitted by March 28th. I am going to now make a short announcement about a workshop that the Delta Science Program is hosting 1 week from today. The salinity management workshop is going to focus on human dimensions of those actions and for actions of salinity management. And this is a 2-day virtual workshop that aims to discuss tools and strategies, identify knowledge gaps, and build shared goals for adaptively managing ocean water salinity intrusion into the Sacramento-San Joaquin Delta. So there's 2 days. If you're interested in both days, I highly recommend that you register for both days. They're different. Day 1 focuses on trade-offs involved in salinity management actions and who is impacted. Day 2 has a focus on modeling results, comparing different management actions and amounts of sea level rise. The agenda is online, and so is the registration. Again, 2 separate registrations for the 2 different days. And the links are on here as well, if you're interested in pursuing joining that meeting. And that would be the end of agenda item 3, that announcement. So I can take any comments from the room on our agenda item 3. And I'm seeing no hands in the room. So how about any comments online, Eric?
No comments online.
Great. Okay, thank you. I hope we'll have some good attendance just like today. So at that point, we can break for, move into general public comment. And so I will open up to the public to provide comments on general issues that haven't been addressed already in this agenda of today. Any comments in the room? Any comments online? Thank you.
No comments online.
All right, well, that would conclude our public comment phase. And I should have put this up. This is the general public comment period right here. You can also submit Any comments, again, on the CHABs monitoring strategy by email to the address listed here, collaborativescience@deltacouncil.ca.gov. And we are accepting those until 5 p.m. on March 28th to be considered for the final monitoring strategy. And with that, I will, if there's no more comments, I will adjourn the meeting. Thank you so much for attending.